Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Sofia Carvalho, Senior Research Associate from IBET.
Hello, I would like to thank Sartorius for the invitation to be here today, and let me challenge you with a question. What do you expect from a very good bioanalytic to support bioprocess development? For me, it should be fast to be able to cope with the required timings during development. It also needs to have a high throughput capacity, as we need to measure a lot of samples, several fractions from different steps, for instance. It needs also to allow us to analyze in-process samples to be able to monitor the entire process. And moreover, it also should be robust enough to deal with process changes during development and process optimization. Today, I would like to discuss advances in bioanalytical tools for viral samples characterization, and I want to share with you two examples of bioanalytical tools for viral bioprocess samples that we developed at IBET. Before I started, let me just give you a brief introduction about IBET. We are a Portuguese private, not-for-profit biotechnology research organization that brings together public institutions and private companies as partners and collaborators. Our mission is to advance biotherapeutics research and development, and do that by connecting academia and industry. Our vision is to provide biotech solutions globally, and with that in mind, we grew in the past 30 years through worldwide collaborations with an extensive track record of projects, not only with pharma, but also with biotechnology companies. And IBET is divided in two main divisions, Health and Pharma, which is dedicated to the development of bioprocesses and engineering tools for biotherapeutics, ATMPs and vaccines as well, and also Food and Health, that is more focused on the agri-food industry. And here are the leading R&D areas among these two divisions, ranging from cell line development to bioprocess development of cell and virus-based biologics at Health and Pharma division, here highlighted in blue, and also food safety or nutraceuticals on the food division, here highlighted in green. And the expertise within IBET allow us to have in-house groups working in distinct areas, tackling challenges at different stages of the bioprocess development. And these core units are supported by the late-stage R&D and bioproduction unit, which is focused in supporting projects or even IBET labs that demand scale-up productions or specific protein production processes, and also by our analytical service unit, which is GMP certified. And IBET has a long track record on the development of bioanalytics, and we are investing a lot on expanding our portfolio of tools to characterize biologics and also support the bioprocesses. We have analytics for cells, assuming that they are our product of interest, as for the case of cell therapy, but we can also look at them as our factory for the production of our products, which is the case in the examples I will show you today. Here, we need to look at cells, identify and characterize them to assure we are producing a high-quality product. And to do so, we need to use a portfolio of analytics ranging from the basic cell identity markers, morphology and structural tools, to secretome and receptome analysis, and also characterization of different omics profiles. When we are looking to viral particles, for example, as the product, the needs in terms of analytical tools are different. Of course, we have to have similar tools like electron microscopy to look at viral structure or characterization of omics profiles. But in this case, we should look as well to viral titers, biologic activity, kinetics, and so on. And these are some examples of the bioanalytics that we are using here to characterize these viral particles. Today I will be more focused on analytical tools for virus quantification, but we have several for upstream, downstream processing monitoring, as well as modeling. At IBET, we have been using animal cells for the production of several virus and virus-like particles for vaccine applications. And these particles can be very different in terms of size, in terms of the presence of an envelope, and so on. And understand all of its characteristics, it's critical to a successful design and performance of the bioanalytics, and of course, bioprocess monitoring and optimization. I will show you two examples today for rotavirus and influenza VLP vaccines. And let's start with the rotavirus vaccine. For the case of rotavirus vaccines and so many other vaccines, there is a demand to improve analytical tools, in particular virus titer measurements. And traditional potency assays, such as cell culture infectivity dose at 50%, also known as CCID50, present several drawbacks, being typically time-consuming, labor-intensive, and also presenting a high variability. Other methods, such as the PCR or fluorescence-based detection techniques, are more sensitive, allowing an early detection, in this case, only a few days, and the quantification of the infected cells. Nevertheless, these methods are still cell-based assays, also presenting high variability and lower throughput. And these are not compatible with analysis of samples during bioprocess development. And biolayer interferometry analysis, usually known as BLA, can contribute to overcome this type of challenges. BLA is a labor-free technology that measures biomolecular interactions in real time. It is an optical technology that uses fiber optic-based biosensors, and these biosensors are coated with different chemistries for different ligand immobilization. And the system monitors biolayer thickness shift over time. And we are measuring a binding response over time as a function of an increasing biomolecular density on the biosensor surface. As you can see here, we have a signal when, for example, an antibody binds to the biosensor, here in blue, and then you have a wavelength shift when the analyte binds the antibody, here in purple. And this wavelength shift is what we measure, and it is represented by these binding curves that we have here on the right. And importantly, only molecules that are binding to or dissociating from the biosensor are able to cause a detectable signal. And BLI analysis can be performed using an Octet platform. The Octet provides a higher throughput. In fact, we can do protein quantitation in less than 5 minutes, and it is also able to process in parallel up to 96 samples. When we are using Octet, we are doing a real-time analysis as the binding interactions are continuously monitored as they happen, and it is also compatible with crude samples, so the analytes can be measured even in crude unpurified mixtures. And as I previously mentioned, it does not require any sample labeling. The first case study we will be discussing is the development and implementation of a high-throughput method for in-process samples that uses this BLI technology for the quantification of a rotavirus in the Rotarix vaccine at different stages of vaccine manufacturing. And this work was performed in collaboration with GSK. So the method explores different strategies based on different proteins of the rotavirus. Strategy 1 uses the binding of a VP7 protein, a capsid protein of the rotavirus, to an anti-VP7 antibody. And strategy 2 uses the spike protein, VP4, and its interaction with receptors that are present in the cell. And for different virus, similar strategies can also be explored. So here we have an overview of the two strategies that I just mentioned. So you can have a functionalized protein A biosensor for the antibody or a streptavidin-1 for the biotinylated receptor. And briefly, we have a baseline step to hydrate and calibrate the biosensor with our buffer. Then we have the loading of the antibody or the receptor, depending on the case. And then there is a new step of a baseline to guarantee that the molecules that are not attached properly to the biosensor are removed. And the next step we have is the rotavirus association to the antibody or the receptor. And this, as I mentioned, results in a shift on the wavelength, and is this difference that we are measuring here. And depending on the type of sample, if we are using a crude or a final product sample, there are impurities that can be removed during the dissociation process. So to implement this type of methods, one of the first parameters to consider is to select the best biosensor or ligand strategy. And to select the best approach, several parameters should be evaluated, meaning the binding magnitude, the association profile, or we should take in consideration how we will do the data analysis of the processed kinetic data. If we are using an initial slope, or if you are looking to the response at the equilibrium. And in this case, looking to the example of the glycoconjugate receptor, we can observe that we have different profiles depending on the strategy that we are using. And for this case of the receptors, we have a binding profile presenting a three-step binding transition, as you have here highlighted in blue, yellow, and green. And this is not the standard association curve behavior that is usually reported for this technique, and can suggest that we are having some conformational changes on the receptor during the association step. On the antibody case, the profile is very different, as you can observe. So the binding magnitude of the VP7 protein to the monoclonal antibody is higher than the one that we obtain with the receptors. And it is possible to achieve a stable steady state response. And in this case, no multiple transitions were observed. So considering these parameters evaluated, we proceed the assay development with using the antibody anti-VP7 strategy. Also, to optimize the binding response obtained, it is critical to define the optimal antibody loading concentration. And to do so, you should screen several, in this case, anti-VP7 concentrations and measure the binding responses. In this example that we have here, biosensor saturation was observed for concentrations above 15 micrograms per ml. And for concentrations ranging from 2 to 15, the binding response was dependent on the concentration that was, the antibody concentration that was being used, with the 2 micrograms per ml concentration presenting the optimal, which is the highest binding value. And this is critical to avoid interferences. And this fine-tuning of the optimal antibody concentration should be considered for each antibody that you are using and for each protein A biosensor lot that you are using to avoid lot-to-lot variations. Another point that also needs to be considered is how you avoid and how you evaluate if you are having or not non-specific binding. In this case, non-specific binding of rotavirus particles to the octet biosensors we are using. And it is necessary to block the biosensor and measure this possible virus association to naked biosensors. Naked biosensors are biosensors that were not loaded with the anti-VP7 antibody, and we should compare this association with one biosensor which we loaded the antibody. And as you can observe here in this figure, no binding occurs to the naked biosensor, here highlighted in yellow, meaning that the blocking step was efficient and that the binding that we are observing is in fact specific for the antibody we selected. Additionally, it should also be evaluated the antibody binding specificity to the whole virus particle compared to a free VP7 protein that may be also in our sample. And the binding value observed in this case for the monomeric recombinant rotavirus VP7 protein that we measured, and here is highlighted in yellow and in a grayscale. And in fact, the binding value that was observed is negligible for all the loadings of this free protein, even when we use a VP7 coated biosensor or the naked biosensor. Moreover, we can also observe that there is a dissociation of these VP7 free protein samples from the antibody or the biosensor, which is characteristic of a non-specific binding. And on the contrary, for the whole virus, here highlighted in yellow and with the blue line, what we can see is that when you have the rotavirus particle, it presents a higher initial binding rate and also a higher binding response. And no dissociation profile was observed. We have here a flat blue line. And these results confirm that, in fact, the binding that we are observing is specific for the whole virus particle. So we need to have the bioactive form of the virus to have a binding to the VP7 antibody, one that we can visualize here in the octet and the BLA analysis. So this validated the use of this assay in the evaluation of rotavirus particles potency. We used our reference standard, which was a sample from the bioprocess that was previously quantified by the CCID50 method to establish a calibration curve. And well, the assay presented a linear response range within 2.5 10 to the 7 to 1.10 to the 8 particles per ml for the range of concentrations that we evaluated. And the LODs, the limits of detection and limits of quantification calculated were 2.5 10 to the 6 and 7.5 10 to the 6 particles per ml. So also the interassay precision was evaluated by analyzing the response of our reference standards that was loaded on 3 independent runs. And we have a coefficient of variation of 4.1%, confirming the high precision of the assay. And the precision that was observed for the methods, combined with the real-time analysis capability and higher throughput of these methods, makes this approach highly promising to cope with the current needs for in-process samples analysis. Moving to the evaluation of the quantification method itself, several in-process samples were analyzed, and the same samples were also quantified using the CCID50 method for tighter comparison. And as you can observe, this BLI method allows the quantification of samples from distinct steps of the purification process, as well as vaccine batch-to-batch comparison. And the tighter values that were obtained for both methods presented similar values, which were confirmed by the Bland-Altman analysis. The Bland-Altman plot can be used to compare two measurements by two different methods. For example, one new method, as this one using the BLI, and a gold standard. For example, the CCID50 that we are using to compare. And the mean bias that is obtained, which is the average difference between the two methods, is small. It's minus 0.02. And all of our measurements fall between the limits of agreements that we have here in the dashed line. So this means that the Bland-Altman analysis do not show any significant bias. So our BLI method is not over or underestimating the rotavirus titer when we are comparing the values with the CCID50 method. So we were able to develop and implement a new tool for rotavirus quantification that made vaccine sample analysis much simpler and faster, which makes this method well suited for bioprocess development. So the methods described here has the potential to improve vaccine process characterization, excelling manufacturing pipelines not only of vaccines already in the markets, but also new ones. And importantly, it can be adaptable to other rotavirus serotypes, other types of rotavirus vaccines, or other viral particles-based products. And in fact, we can adapt and optimize it also for virus-like particles. And that's the second case study I would like to share with you, the development of a method for influenza virus-like particles quantification under the scope of EduFluvac European project. And the aim of this project was to develop a universal vaccine using multivalent particles, combining multiple influenza hemagglutinin or neuraminidase proteins. And the concept of this universal vaccine relies on the display of these multiple HA and or NA epitopes that are either of different strains, of different influenza strains within one subtype, or from different subtypes, covering as much influenza virus diversity as possible on the surface of the scaffold, and inducing the generation of a broadly reactive antibody with capacity to neutralize upcoming either seasonal or pandemic influenza virus strains. And here we have just an example of a pentavalent VLP which has 5 different HA proteins. And one of the challenges that influenza bioprocess people are facing is accurate and fast quantification of hemagglutinin protein. And the current approved analytical methods, which are RID, single radial immunodiffusion, and hemagglutination assay, are impractical for inline process monitoring, mostly because they are time-consuming. The VLP concentration in crude samples generally falls out of the range of detection, or because they have a lot of interference. Also, these methods are more accurate when you are using influenza virus, and in this case, we were using virus-like particles. And for instance, for the hemagglutination assay, we need to use fresh red blood cells that are highly unstable. So it was necessary to develop functional process analytical techniques that can be applicable at every stage during the production of these VLPs. And for influenza virus quantification of in-process sample analysis during the bioprocess development, we usually use hemagglutinin protein. And hemagglutinin protein plays an important role in host tropism, since the viral receptors vary according to the host, impacting infection and replication of influenza virus amongst species. For instance, avian influenza viruses engage preferentially alpha-2,3 linked sialic acid moieties, where human-adapted ones have a higher affinity to alpha-2,6 sialic acids. And in this case, the strategy used in this assay development and implementation was similar to the one used for the rotavirus case study. However, this one explores the interaction of hemagglutinin protein with the biotinylated sialic acid receptor instead of the antibody. And these receptors are bound to a streptavidin functionalized biosensor. In this case, as we want to compare results from our BLI methods with the hemagglutination assay that uses chicken red blood cells, and we are dealing with hemagglutinin proteins that have a higher affinity to human cells, we use the mixture of both 2,6 and 2,3 sialic acid receptors. So here we have the quantification results for the in-process samples, and as we know, depending on the downstream processing step, samples will have different purity levels, leading to different response behaviors. And in this case, all the analyzed samples were grouped based on the DSP step that they belong. And these black lines that we have here in the graph represents the error range that is associated with the hemagglutination assay. And the overview of our results show that the majority of the samples quantifications are between the error range, being in accordance with the hemagglutination assay. And this panel here is a zoom of that initial area of the first panel that I showed you. And in this case, we are looking specifically to clarified samples that are more challenging to quantify because the samples from the clarification stage contain several culture media compounds that can possibly interfere with the concentration analysis. And this, importantly, this is already observed as well in routinely laboratory hemagglutination assays. So it's a common issue. And for that reason, it's more common to have small differences in both quantification methods that we are comparing for these early stage samples. As the downstream processing advances, the purity increases and the HA concentration values that are calculated using the BLI assay became closer to the ones acquired with the hemagglutination assay. And also, to evaluate specificity, several strains from the same subtype or group were analyzed. If you remember, we have for this EduFluvac project multivalent VLPs with several HA proteins from different subtypes or groups. And we use several strains from the same subtype or group analyzed against the same calibration curve. And these experiments were performed for H1, H3, and B subtypes for several mono and multivalent strains. And here is an example for the H1 subtype group. Strains from this group were quantified using only one H1 strain as a calibration curve. And in particular, it was an ultrafiltrated retentate sample that was used as calibration. And then we used 3 monovalent, 1 trivalent, and 1 pentavalent VLP sample to be quantified against this calibration curve. And the results that we have here show that subtype and group quantification is possible with only one sample out of the error limits that we had. And this was also observed for H3 and B group samples that also contain 3 and pentavalent VLPs. So we were able to develop a fast and robust assay for HA quantification, and this one does not need to use red blood cells and uses instead stable and homogeneous receptors. And as for the other case study, this one also enables to have a bioanalytical tool that is compatible with the time constraints that are faced during bioprocess development. And in fact, having this analytical tool implemented supported influenza VLP bioprocess development for all the multivalent VLPs produced. So coming back to the characteristics of a well-suited bioanalytical tool, BLI analysis using the Octet platform allowed us to have a faster analysis. For the rotavirus example, we moved from days of waiting to the results to minutes, and the influenza method enables a 15-minute response instead of 1 hour. And these factors, as I mentioned, make this method compatible with the time constraints that we have during bioprocess development. Moreover, using the Octet, we increase the throughput, being able to analyze a much higher number of samples with less resources and costs associated. And as the method presents a wide linear dynamic range and a limit of quantification suitable for quantification of in-process samples, we were able to analyze the entire bioprocessing, ranging from harvest to the final product, purified product. And at this point, we have an influenza label-free quantification tool that spares us to use the red blood cells, and not only is suitable for several mono or multivalent influenza strains, but also presents subtype and group specificity. And for the rotavirus vaccine, we were able to quantify the entire bioprocess and also do a batch-to-batch comparison. That's what I wanted to share with you today. I would like to thank all the IBET members involved in both projects, in particular Mafalda Diaz and Mafalda Moleirinho, and the Animal Cell Technology Unit, our collaborations, in particular at PAL Life Sciences, that supported the influenza project, and GSK, who collaborated with us in the development of the rotavirus method, the funding, and again, Sartorius for the invitation to be here today. And before I take any questions, let me just leave you with a take-home message on how the development of new analytical tools and retooling of methods that are already established in other fields are much needed. To optimize product characterization and process understanding, to excel biologics development and manufacturing pipelines, and also to support the increasing demands on bioanalytics for these biologic products. And as we saw today, Bioanalysis using the Octet platform can be considered an excellent tool to do it. Thank you.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions now. So our first question is, were the octet methods stability indicating?
Hi, so yes, the octet is a technique that we can use to develop stability indicated methods. But even though we didn't show today any results on that, it's already established, for example, for influenza molecules as well. So we can, for instance, inactivate the antigen of the influenza virus through it or other forms of denaturation, and then we can show that this denaturated form does not bind to the immobilized ligand. So yes.
And what are the advantages of antibody versus receptor binding, and when to choose one over the other?
It depends on the viral particle and on the method you want to develop. So antibodies are more specific than the receptors. And if you want to measure a very specific binding, it will be easier to use an antibody. However, sometimes we don't have the antibody available. For influenza virus, for example, if you want to develop a more universal method that is suitable for several strains, groups, and valencies that can cope with the viral antigenic shift and drift that is specific for this virus, using antibodies is more problematic, as you will need several antibodies instead of only one. For the case of the rotavirus that we reported here today, the binding observed to the receptors that we evaluated was not suitable for the method development. So as we are only dealing with one serotype and we have the antibody available, it was a viable option. So if you have a viral particle that has a more conserved structure, the antibody could also be an option.
All right, our next question is, how are you sure that what is being measured is the virus and not the protein on the octet?
Yeah, so that is one of the points that we showed today. So to observe a binding, you need to have a wavelength shift that is measurable. And we evaluated both recombinant HA protein and VP7 protein, and in fact, the protein itself is not large enough to cause a measurable wavelength shift. So the binding that we are observing are in fact from the bioactive form of the virus or virus-like particles, and not for protein that is in solution.
And to what degree are the viruses and virus particles what are being made or have gained unwanted access to the product being made? What is the balance between unwanted and wanted viruses and VLP?
So I'm assuming you are asking more about the purity and if we have other species that may interfere with the method, and if the method is specific. So in fact, we are measuring in process samples. So we will have impurities in solution that can interfere with the method, and we show that. But we know that the assay is specific for each virus evaluated because the binding of that virus or virus-like particle is specific for either the antibody or the receptor that we are using. And we also confirm that the binding of the virus to the biosensor itself using the naked biosensor is negligible. So impurities like host cell proteins can slightly bind to the receptor or the antibody, but then they will dissociate as well in the dissociation step. And that will happen as well in other orthogonal analytical tools. That's why it's more challenging to quantify in process samples. As we already discussed, free protein do not present a measurable wavelength shift. So in fact, we are measuring only the virus or the virus-like particle.
All right, and could you advise how to handle of VLPs on the octet and recommend some best practices?
I think the settings that you will choose on the octet will depend on the virus-like particle that you are using. For this case, for the influenza virus-like particle that we have, some of the multivalent ones were very unstable. So the settings that we needed to use were different from others that we already used with other type of virus. So for these virus-like particles, room temperature was the best option, and we needed to use the maximum agitation as the particles, especially the particles that are in process samples, tend to settle to the bottom of the 96 well plate.
And what shake speeds were used for analyzing the virus titers, since the purified proteins did not show any binding at the antibody or reagent concentration used?
So we use 1000 for both loading and association.
Okay, well, it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Sophia, who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what blocking reagents were used for blocking HCPs or in-process material?
Yeah, so the blocking reagent that we use is the one that is provided by Sartorius, is the kinetics buffer.
All right, great. Well, thank you, Sophia.
Thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Bye-bye.